Hey guys, so I wanted to talk today a little bit about spermatogenesis. We just had a class on this, and I wanted to kind of walk you through the pictures that we drew on the board as an extra resource. So first off, spermatogenesis is the production of sperm. These are gametes produced by a male animal. Typically, the situation for almost all animals is that the males produce a lot of small and motile sperm, while females produce a small number of larger stationary eggs. And the idea here is that by producing a lot fewer eggs than sperm, organisms can kind of control how many offspring are produced in the next generation. Because like the more eggs that are produced, the more potential offspring. Now, for some organisms, that's exactly what works the best. Like in sea turtles, for example, they have over 100 eggs or so in each nest, and that's great because they have a high rate of juvenile mortality. Very few of those sea turtles make it to maturity, but many other animals want to produce fewer eggs because they don't want 100 offspring. Many other animals exhibit a lot higher degree of parental care, so they want to produce fewer offspring that they can care for and ensure their survival. All right, so that's kind of the general rule. Lots of small sperm, few larger stationary eggs. Another thing is that the average male produces a lot of sperm every day, and this goes for humans and other animals. The average male human will have 250 million sperm in the ejaculate, which is a huge number. There's like 350 million people in America, so this is like the population of a big country. All right. And that means that lots and lots of sperm are produced every day. Typical male produces like 150 million sperm every day. And about 400 million of those sperm can be stored in the testicle. All right, so let's switch over to the picture of the board here. All right, let's try this. There we go. So that's kind of what we talked about earlier. Lots of sperm per ejaculate. Here's a drawing of the male testicle from the lateral view. To the right is the anterior side, left is posterior. What you're gonna see, you see this blue oval, that's the testes. And inside the testes, you have these brown little tubes. Those are called seminiferous tubules, and these are the tubes that produce new sperm. All right, so new sperm are produced in these seminiferous tubules, but when they're first produced, they're not ready to fertilize an egg. They're definitely not mature, so they will slowly make their way out of the seminiferous tubules. If you follow these little purple arrows, they enter into this really tangled up bunch of brown tubes called the rete testes, and eventually they emerge out of the testes to enter into this blue or this aqua green tangled up tube. That is called the epididymis. The epididymis is long. It's about 20 feet long, and the sperm will make their way slowly down to the bottom of the epididymis. That's called the tail, where they are stored. All right, so that's where they're stored. And again, the average male can store about 400 million of these sperm in the tail of the epididymis. When it's time for these sperm to leave during ejaculation, that's when you'll get these peristaltic contractions of this red tube called the ductus deferens or the vas deferens, and they propel the sperm into the pelvic cavity, where they get mixed with other fluids from the prostate gland, the seminal vesicle, that's the semen. Together, the semen and the sperm make up the ejaculate. So that's kind of the overall process here. Another thing, these Leydig cells, these little pink dots that are arranged around the seminiferous tubules, those Leydig cells produce testosterone, which has a lot of effects on the male body, but one role that it has is to stimulate spermatogenesis. Without testosterone, there are no sperm being made. All right, so over here, this is what I really wanted to focus on, because this is a zoomed in view of the wall of the tube. Imagine we took one of these seminiferous tubules and we cut it in half. This little brown line, that represents the outside of the tube, and towards the left, like right here, that's the center of the tube. Now, along the outside periphery of the tube, you have these little blue cells called spermatogonia. These are germ cells that will undergo mitosis, right? And they divide towards the inside of the tube. Now, these new spermatogonia are then gonna enlarge. See how they get bigger? And they're gonna become a new name. They're gonna enlarge and they're gonna become the primary spermatocytes. These primary spermatocytes are gonna enter meiosis 1. Now remember that meiosis is the process of gamete formation, where sperm and egg are produced. So that's essentially what's happening here. These primary spermatocytes are then going to finish meiosis 1, and they're going to enter into meiosis 2. Once they go into meiosis 2, they become the secondary spermatocytes. So those are the 4 cells that are slightly more towards the center. After they finish meiosis, then they become these small cells called spermatids. Now keep in mind, according to meiosis, you have 1 spermatogonia that undergoes meiosis to produce 4 daughter cells. These 4 daughter cells are called spermatids. Those are the little cells on the right. As you can notice, they really don't look like sperm. And in fact, the next step of their development is to turn into these purple spermatozoa. They undergo a pretty dramatic transformation where all of the extra fluid and cytoplasm is ejected out of the spermatid, and they start developing this completely new shape that looks like what you're used to seeing with the sperm, okay? Then these spermatozoa are released into the lumen or the inside of the tube, where they begin their journey all the way to the epididymis. That journey takes about 3 weeks from the time they leave the seminiferous tubule, or they enter the tube until they get to the tail of the epididymis. All along that time, they're developing, they're getting ready to be able to fertilize the egg. Now, if we kind of summarize the whole process here, what we had a spermatogonia. It enlarged, it entered meiosis to become the primary spermatocyte. Once it finished meiosis 1, it becomes the secondary spermatocyte. Once that secondary spermatocyte finishes meiosis 2, it becomes the spermatids. Those spermatids develop to become spermatozoa, which are released into the seminiferous tubule. Now, just as a quick overview of meiosis, meiosis is a process in which gametes are made, sperm and egg. And these sperm and egg have a haploid number of chromosomes. That's half the number of chromosomes compared to all the other somatic cells of the body. In humans, our somatic cells have 46 chromosomes. They exist as 23 pairs of 2. Sperm and egg, they must contain 23. And it's just a math problem. It's a numbers problem, because if sperm contain 46 and eggs contain 46, 46 plus 46 equals 92, and that's way too many. So we need to half that number so that when fertilization occurs, the correct number of chromosomes are achieved. All right, so imagine we had a cell that only has 2 pairs of chromosomes. So 4 chromosomes total. In meiosis 1, the first thing that will happen is that these pairs will join together. See how in this picture you have the 2 pairs, the blue chromosomes from mom, the pink chromosome came from dad, they join up. This joining is called synapsis, and during synapsis, the 2 chromosomes are really like twisted up together, right? So textbooks like to show them as side by side, but they really are like twisted up. This allows the process of a crossing over to occur. And here, what will happen is of each pair, a little bit of genetic material from one chromosome is swapped with the other member of the pair. This is really important, and it must occur in order for meiosis to proceed. The location where these 2 chromosomes join is called the chiasmata. So after that happens, meiosis 1 is going to complete. And during meiosis 1, the members of each pair are separated. Okay, and they go to 2 different cells. That's pretty much the whole goal of meiosis 1. That means that since we've separated the pairs, these new 2 cells are haploid. That's what the N means. They have half the number of chromosomes. In meiosis 2, the chromosomes are then split right down the middle at the centriole. If you remember, when we talk about chromosomes, each side of the chromosome contains identical genetic information. So meiosis 2 just pulls the 2 sides apart. This is going to result in 4 daughter cells that each have a haploid number of chromosomes, that each have 2. But if you zoom in, you'll notice that because of crossing over, not one of these cells contains identical genetic material to another. They're all unique. See how that pink chromosome has a little bit of material from the blue one? That crossing over ensures that each gamete that results from meiosis is unique. In fact, there's an infinite number of genetic possibilities that are produced through meiosis, leading to an infinite number of genetic, combinations of sperm. No 2 sperm or eggs are alike. All right, and that's about it for that. Next lecture, I think I'm going to talk about the structure of this sperm, which we have right here.